I'll confess, I'm a relative newcomer to the CRISPR field. I was working in the lab for many years on problems involving fruit fly development. In fact, how you make these wing patterns and so forth. When a very talented graduate student joined the lab, and he's an artist as well as a fine scientist. Both of these are drawings by him of a fruit fly with a PCR machine in its back, where it's active on one side but not on another, and I'll get to why that's an analogy we pursue. And then the Vitruvian mosquito, right? The famous proportional diagram of a human being by Leonardo da Vinci, which is kind of appropriate because Valentino Ganz, the person who did this, is in fact of Italian heritage. And so I joke around a little bit at the time that when Valentino came up with this little idea that there may have been, for the problem he was working on in the lab, there may have been 4 people in the world who cared about what he was doing. Himself, me, and perhaps 2 other people. And the reason I bring it up is that it's from these kind of bizarre little corners of research sometimes that some new ideas of consequence potentially can come, and how important it is as we go forward to preserve the fundamental concept that came out of post-World War II science in the US, which is to support basic research. Critical in my view. Okay. So I don't have to go over this here. And I will just go very quickly through it. We all know how that works. And so, of course, you get a double-stranded break. You have to repair it, or the cell is not happy, dies. So you either have NHEJ, which is error-prone, versus HDR. Now, I don't want to dwell on this, and at the end, if you want, I'll be happy to go through this point in more detail. But this is a really gross oversimplification. I hope you all understand that, right? First of all, NHEJ is not particularly error-prone. Okay. The reason it keeps coming up as error-prone is because if you keep cutting things over and over again, eventually it makes a mistake. But actually, when people have looked on a per-cycle basis, NHEJ is just about as good as HDR. So it's not really true. Okay, number one. Number two, this is a very generic diagram. And I have another one that I won't impede you with, but if you want, we can go through it at the end, which compares how does this actually work in the germline versus somatic cells, and it's completely different, okay? It's about diametrically opposed different, what happens in the germline versus the somatic cells. Just think about it. What does a somatic cell want to do? A somatic cell is a happy little thing. It's cooking along. It wants to have a, do a little replication to, grow or to heal a wound or something. The last thing it wants to do is have its progeny be any bit different from it. They want to have the daughter cells to be identical to them. Identical, right? So what does it do? You go through replication, and then at the situation where you have 4 strands, you repair from the identical sister chromatid because you want to preserve genome integrity. Now you move across to the germline. It's a completely different thing. It happens at the situation where you have 4 copies of DNA again. But now what you're trying to do in meiosis is you're trying to scramble the information from mom and dad. You want to go back and forth. You want a little piece of dad's chromosome, a little piece of mom's chromosome. You suture them together and you give them to your F1. Yeah, your sons or your daughters. Totally different. One preserves genome integrity at the purpose of it. The other one is to scramble genetic material. So in meiotic HDR, you copy from the homolog. And in fact, it's not like there's DNA repair that's being foisted, damage that's being foisted on you by X-rays, chemical mutagenesis, or all kinds of other unpleasant things, replication errors. It's actually SPO11, an enzyme, that actually goes and cuts it's a type 4, topoisomerase that makes a double-stranded DNA break on purpose. So it's intentional DNA cleavage. Not only that, but there's a enzyme cassette that builds on the SPO11 and builds a protein complex, and then that protein complex eventually recruits the RAD51, which is what, as I'm sure you're all aware, that's what triggers the HDR pathway and goes down that pathway. So this is a gross simplification. That's all I want to say. All right. Nonetheless, this is coming up now with hindsight on this, and when Valentino came up with this, we had no idea. We just thought naively that this was hopefully going to happen, okay? So we didn't really know what we were doing, honestly, at the time when this idea came up, because we weren't part of this field. It's a lovely field, it's a great field, transformative field, but this wasn't what we were doing at the time. Okay, so what Valentino's idea was, well, he was frustrated by a problem going into a different insect where, although we had the CRISPR tools that we had borrowed from others who had worked them out for fruit flies, people who are pioneers in that area, we were just using them as standard users, and Valentino had gotten it to work really well. So we were making deletions and studying our cis regulatory elements that were all of our interest about how you make a wing vein. Again, not exactly something that's going to send ripples of excitement through the audience, right? But nonetheless, that's what we were trying to do. But the idea is we're kind of evo-devo. We're kind of evo-devo folks. We want to know, how does it work in a different organism? And eventually, could you take control over evolutionary processes take information, be informed by evolutionary processes to actually, from first principles through sort of synthetic biology, create new kinds of structures at will, by design. That's the kind of idea. That's the sort of thing we're hoping to learn from all of these seemingly esoteric experiments. But anyway, the problem was that in this distantly related fly, about 160 million years diverged from Melanogaster, those tools weren't working very well. He took the U6 from that species, put the guide RNA under there, put the Cas9 in there on a plasmid under the control of the VASA sequences, which is a germline promoter sequence. And it just, wasn't really doing very much. We weren't getting the mutants that he wanted to get. And so in a moment of, frustrated, brilliance, or whatever you want to call it, moment of, the light going on, he said, well, how about if we could just make that characteristic that we want to see, which in our case is the loss of a wing vein, okay? How about if we could make that characteristic just appear in the first generation? Because as you know, typically, mutations are recessive. So you have one mutation in mom, one mutation in dad, you cross them, and then the kids, they have the defect, because that's what you have to do if it's a recessive mutation. Well, if you're not very efficient at generating them, you're not getting biallelism 90% of the time, which of course you can in fruit flies now, just like you can in these other organisms. But if it's a different organism, you don't have that yet, and let's say it's 1 in 100 or 1 in 1,000, well then, you just do the math. It's 1,000 times 1,000, that's 1 million. You'd have to look through 1 million animals before you found the recessive phenotype. That even for fruit flies, that's asking a lot. Okay, so he was frustrated, and so he said, how about if we could just do it in one generation? So he came up then with this idea where you put the Cas9 and the guide RNA that are cleaving at a certain point in the genome, and then flanking this cassette, you put the homology arms that have endpoints that directly abut the cut site. So this homology arm, this cut site here is here, this base, to the base of where it cuts. And so now the idea is, well, it'll happen in the DNA repair process, again going through HDR, and now thinking of this as a little homolog chromosome. This is all retrospective thinking, right? Because in fact, this probably does not happen in the case of where he had wanted it to work, which was in the somatic cells of the body. In the somatic cells of the body, this probably is not what's going on. But we'll get back to that later. Anyway, but in the germline, fortunately, it does. So in the germline, what happens is, you repair this break by copying down into here, taking this genetic material in, and then back out through here. And that results then in the incorporation of the Cas9 guide RNA exactly into the place where it's designed to cut. Then you make the protein genetically, from the genome, and now, of course, you've got huge homology arms that go the whole chromosome length. You cut, and again, it's the same thing. You repair the break by copying through here and then back down, and then boom, you've got it's biallelic, okay? And so here's a little cartoon that he put together. Artist that he is. That's a brown fruit fly, which is the normal color of fruit flies for those of you who haven't had the pleasure of seeing one recently. And that's a blonde one, a yellow, so-called yellow mutant. And then this little cassette disc gets copied back up in there, just like I'm saying. And now the animal is yellow. Now again, it's an interesting point to ask what's going on in the body of the fly versus what's going on in the germline of the fly. At the time, we were thinking that they were the same thing and that we would use the germline transmission as a proxy for seeing how well the thing copied, because it's kind of hard to actually look in the cells themselves to see if it's copying in the cells. Now, we're actually creating a construct that's going to allow us to do that, and I'm looking very much forward to seeing how that works. But in the meantime, what we had done is we had just used the propagation genetically. So you know what happens if you take a mutant versus a wild type, and my apologies to the true fly aficionados here who know that the yellow mutation is on the X chromosome, but this is kind of the generic thing where you have a mutant and a wild type, and the kids get one chromosome from each one. So at this stage, the heterozygotes, again, have 50% inheritance of the allele, the mutant allele, and 100% wild type. That is, again, it's a recessive mutation, right? That's what everybody has seen since they were in 7th grade. No? 7th grade. 7th grade genetics. Okay? Now, the same thing starts off with Valentino's construct, which he called the MCR. And that's a name that, in retrospect, we might have given it a different one. But it's called the mutagenic chain reaction. And it was meant to be an analogy to the polymerase chain reaction. Because the polymerase chain reaction, every cycle, the DNA gets doubled in a test tube, and here it gets doubled every cycle, but in vivo. So that was the idea, and we were studying mutations, Anyway, for better or worse, that's what we called it. And so you get one of each, but now the difference is that one converts the other, and then what happens is you have 100% inheritance of the allele and 100% mutant. And that's very, very different. It's imagine if you can just consider the analogy again between blonde and dark-haired traits. Typically dark pigmentation is dominant over blonde pigmentation or light pigmentation because it's the presence of pigment, right? So if true breeding dark organism mates with a true breeding light organism, then the kids are all dark, and then only in the grandchild generation can you get a quarter that would have the recessive trait, the light trait, right? But now imagine here what happens is you have, like, say, a blonde person that would marry, say, a dark-haired person. All their kids would be blonde, all their grandkids would be blonde, all their great-grandkids would be blonde. Now that's pretty different, you'll have to admit, to the standard form of inheritance. And so this was the data from that first paper. Since this trait is on the X chromosome, one has to consider it in females that have 2 of them. And of 436 progeny in that first test that were looked at, all but 6 of them, or 8 of them were blonde. And that was a 96% transmission rate, because you have to account for the fact that by chance alone, half the time you'd inherit the allele. So of the other allele that you would have that would be the different allele, what fraction of that time did it get converted? And that's about 96%. We've since redone it with much larger numbers, and this is probably a bit of an underestimate. Okay, so then based on that kind of initial finding, Valentino and I sat around for a while, and we considered all the other possible things in principle one might be able to do with what we're now referring to as active genetics. And by active genetics, I mean sort of using this kind of CRISPR technology to engineer things so that in a Cas9 dependent way, all the progeny can inherit a given trait. And initially the trait is a transgene, but again, speaking to some of these interesting characteristics that you might want to just make point mutants, I'm going to give an example at the end, toward the end, of what we call allelic drive, where we can move a single nucleotide change throughout a population, in principle. And then, of course, because these systems are capable of being transmitted to all the progeny, I'll start off by talking a little bit about gene drives, since that's actually what's acquired more of the attention from all this technology than anything else. How it can be applied in principle to schemes to control pests like mosquitoes and diseases that they cause. Because there's concern about those things running rampant. There also are neutralizing drives. I'll talk about erasers and an alternative form that can function as we call chasers, which are elements that have the guide RNA that's inserted at the site where the cassette is put into the genome, but there's Cas9 is provided just in a Mendelian sense. So if you can think of a full drive, you could sort of think about it as a car where somebody has actually stapled the gas pedal down to the floor, so you can't stop it. It just goes, whereas the chaser is one where you have freedom to release the gas pedal if you want, so you can drive it in a more careful and conditional way. And then these copycat elements, which are these elements that, again, these guide RNA-only elements that are useful for transgenesis and, in principle, useful for combining very complex multiple traits, and would be, I think, very helpful in polyploid organisms in particular. So I'm going to start off by talking about gene drives, and my good buddy and colleague, Tony James, who has spent his entire lifetime trying to develop mosquitoes as a genetic system to combat malaria. And so he's been dedicated to this. He's a member of the National Academy, a very serious, conscientious person who has indeed had experience in field trials in Mexico, where he has tried to use transgenic mosquitoes as a sort of insect sterile technology to combat dengue. And in fact, the Oxitec mosquitoes that have made a lot of noise, again, because a company that did not necessarily have the greatest skill in communicating with the public, has run into a lot of trouble in Florida trying to implement this technology down the road. But Tony himself has always been very, very careful, tried to make sure that in any such situation where he's proposing to genetically modify a population of mosquitoes, that's done in close consultation with regulatory bodies, government bodies, the local people who are affected, all the stakeholders. And he, in fact, is considered the poster child of a person for doing these kinds of things right. So this is a person dedicated to this process. And there are two then sort of ways you can go. One, which are our competitors that reside in Imperial College, most of them in London, who are that are sort of the intellectual father, that's Austin Burt, who considered using homing endonuclease genes originally as a system that's very similar for population suppression. And here what the idea is that you take a gene drive system and you purposefully use it to purposefully sterilize or even kill the target mosquito. And so it's a genetic insecticide, very much like with the Oxitec sterile insect. Instead, it's in principle much more potent and much longer lasting. And so there are some advantages, of course, of that, which is that if you take Aedes aegypti, for example, which is a mosquito that's of great concern to us in California now because it's spread as an invasive species, it can carry dengue, Zika, chikungunya, yellow fever. It's not a very nice set of diseases there. And those diseases are not being actively propagated by Aedes aegypti at the moment. It could be. There's a sister species, albopictus, Aedes albopictus, the tiger mosquito, which also can carry those diseases, also an invasive species. Now, if you kill that mosquito, you get rid of all of those diseases at once, right? So if you get rid of the mosquito, that's good because those diseases just go away. An argument can be made if it's like the state of California and this is an organism that doesn't even exist normally, had normally not existed at all in the entire Western Hemisphere, that to get it out of California, since it just came in 2013, it's not going to be getting rid of something that's got some kind of, evolutionary ecological hook into the current ecosystem. So it's unlikely that if you got rid of a mosquito like that, that you do any ecological damage. Now, if you go, though, to other insects or other situations where you could use this thing, where you imagine that the species is indigenous, then you could start worrying about ecological harm. There's just the general ethical consideration of should you take a species and drive it to extinction, even if you don't like it because it's causing disease. That's an ethical consideration one can talk about. In the case of a mosquito, they have very, very few people who argue in favor of them. They're kind of like rats, people just don't like them. But that's a bit of a prejudice, and it certainly isn't a mindset that one wants to take to all other possible situations. And there are lots of ethical considerations that come about from using this technology, and this is certainly one of them. Okay, but from a practical point of view, and mostly since I consider mosquitoes and malaria a really bad thing, and getting rid of the disease an obviously very good thing because it kills about 500,000 people a year and causes sickness in over 2 million. That's very disruptive to families. I think that isn't, in this case, I don't think this isn't the biggest consideration. But the big consideration against suppression is that it's not stable. And the reason is because when I mentioned that gene drive works really well, like 96% of the time, and I'll show in mosquitoes it can go up to over 99% of the time. But still, those few percent that don't, where it doesn't copy, you get a non-homologous end joining kind of allele that then destroys the guide RNA cut site. And so now it's a resistant allele. And sometimes the indels that are generated are in frame so that you restore the activity of the gene you're cutting, but it no longer can be cut. So now you've created in a gene that's an essential gene or in a gene that's required for fertility, a resistant allele that is healthy compared to the gene drive allele, which is not. So it's a formula for failure. It's just not going to work. And now there have been a bunch of papers that have come out that have made a big point about this. And I think the essence of it, when you really think about it, is that for this system to work well, you better not try to, at least if you're just going to use a single version of this. There are tweaks on this where you use a whole bunch of them at the same time. And by that time those resistant alleles arise, it's too late for the organism and you can drive it. Yeah, that's a good question. It's not known. I mean, and it depends probably very much on the ecology and the landscape and the population density distributions. But to a first approximation, if you take a population down below 1%, it's very likely to go extinct. Even below 5% under certain circumstances can do that. On the other hand, what happens very often is you can achieve local extinctions or eradications. Usually you just eliminate them locally. And then what will happen is that they get repopulated by other insects coming in and that comes back. That's the Oxitec thing. So even in the cases where Oxitec has completely eliminated mosquitoes from areas, it has done that in certain cases. They come right back in and you have to do it over again. With a gene drive, though, it keeps spreading. And so it prevents that from happening. And so it could do that. But again, I think it's very unlikely that this is going to work because it intrinsically defeats itself by creating drive-resistant alleles. Now, population immunization, on the other hand, which is what Tony James, my collaborator, has pioneered, uses the drives to modify the phenotype of a population. So the idea is simply to immunize them against the malarial parasite. Do no harm to the mosquito, just make them unable to carry the parasite. So it's stable because it doesn't reduce the fitness of mosquito population, and in principle does the least ecological harm because all it does is rid the environment of the parasite, not the mosquito. And if the mosquito has any use, or a rat even would have any use, then you leave those animals in place. You just don't let them become vectors for the disease that you're concerned about. The weakness, of course, is that then an immunization element must be made for each disease. So if you were going to go about this, for example, for the Aedes mosquitoes, you'd have to make one for Zika, you'd have to make one for dengue, you'd have to make one for chikungunya, you'd have to make one for yellow fever. Now, of course, those aren't difficult to do, right? I mean, if you get one to work, it's pretty much just straightforward to make the others, but then you have an organism that has to at least have for transgenic types of elements. You might be able to put it all into one drive system. It's possible. And so you could take out all the diseases at once that way. And in fact, I think one of the most exciting things about some of the RNA-guided CRISPRs is the idea that you could harness some of the RNA-guided CRISPRs to go after these different kinds of viruses. And that might really allow you just to make a single vector that took out all of those different diseases. An aside here that I won't elaborate on too much, but a design feature that has to do with how these things copy is that you had better not put any repeated elements in them. So that takes out, for example, RNAi types of approaches that have been shown to be successful, for example, in making mosquitoes unable to carry dengue. So people have succeeded, Tony, in fact, himself being part of that study, of making hairpins that can work against dengue. But a gene drive, if you put that on a gene drive, it'll delete itself out by the process by which it copies. Anyway, so what Tony had done is he had spent a good 10 years after getting transgenesis to work in mosquitoes, which already took 10 years, to generate this very sophisticated little device which has a cis-regulatory element that responds to a blood meal taken from genes that are normally activated upon blood feeding in the gut of the mosquito, and to use those to drive the expression of effectors, which in his case are two single-chain antibodies that bind to the parasite at two different stages of its life cycle. One that prevents it from going from the gut transversing from the gut into the body cavity, and the other that blocks it from going from the body cavity into the salivary glands. And together, he had shown that these two antibodies, when expressed in this way, could block malaria transmission 100%. So in animals that were fed malarial parasites, never gave those parasites. Those parasites never got into the salivary glands, and they could not be transmitted. And at the end of the paper, where he had gone through all kinds of hoops to identify fitness-neutral loci in which such gene cassettes could be placed that would not harm the mosquito at all, he ended that paper saying if this were coupled with a mechanism for gene spread, then malaria-resistance transgenes could become a self-sustaining disease control tool. So at the time that Valentino came up with this idea, I asked him, well, what good is this little thing? Because I didn't know anything really about the gene drive field at all at the time. And so he looked around, he said, well, one thing it'd be good for is if it could be potentially useful for driving gene cassettes like this. So I called up Tony and I said, hey, would you like to collaborate? And so he said, sure. And so the idea is that we kind of, Valentino, not me, Valentino would put Cas9 guide RNA motor together with this, and then we'd see what could happen. And so anyway, the long and short of it is that when this is driven, and I have to be very precise here about how it's being driven, when it's being driven through males carrying the genetic element, carrying it across to wild-type females, it gets transmitted to 99.5% of the offspring, which again, when you correct for the fact that would happen 50% of the time by chance alone, that's still almost 99% drive. In this case, it's a very big element, a 17 kb element. So I figured if it got in, it would just park there like a school bus and it wouldn't move. And in fact, it was difficult to get it in. Tony had to screen through 25,000 larvae and found 2 that were positive. Okay, it was very difficult getting this in. But once it got in, I figured it wouldn't move, but I was wrong again, and it did very effectively. And so and it carried that whole cassette, that whole antibody cassette. So it really was a good sort of prototype experiment for showing how this could work. So then, the way this might work is that you say introduce it at 1% of the population. It spreads within 10 generations to roughly 100% by logistic growth. And then that presents a problem, right? Because it could potentially go on spreading. That's what people might be worried about. For example, the Cartagena Protocol, an international agreement that almost all countries in the world sign except the United States, and that's probably not going to happen soon, you're not supposed to have a genetically modified organism cross international boundaries. It's illegal to the international community, minus the US. And so then, what would you do then if you had a country that wanted to use this, but they had a neighbor that wasn't so happy about it? How are you going to make sure that the thing doesn't get across there? It's spreads. So one idea are these sort of elements that we call for reversing this reaction. And this is, of course, a backronym just so we can call it an eraser. And the way an eraser works is the key element is that it does not have the Cas9. So the Cas9 has to be supplied in trans, but it has two guide RNAs that can cut on either side of the drive element. And so they make complexes like this, and they cut on either side, and then they cut that thing out, and now you get you've repaired this break by copying through the eraser sequences, and now you have both alleles that have the eraser, but now there's no more Cas9, so you stop the system. It freezes at that point. And so the idea then is that you have a gene drive that spreads through the population. Once it's spread to a point that you're happy with and you don't want it to go any further, you release the eraser. And then the eraser can drive through a gene drive population with the same logistic kinetics that a gene drive can drive through a naive population, and at the same time replace it. So it drives and replaces. And the advantage is that the erasers could also carry the anti-malarial effectors, and then once they do so, it would sort of freeze the process to the radius or whatever that the gene drive had made it to. Now, of course, at that point, the gene drive would no longer function, so mosquitoes from the outside could come back in that carried the parasite, and you wouldn't be able to get rid of it anymore because you'd frozen the process. So that would be the negatory of that. But at least it's a possible way of handling that kind of problem. Now, I'd like to turn to these things we call chasers. Again, they have their own reason, their own name here. But again, the idea is that it chases after a gene drive. And they're basically updating platforms. And so here would be one drawn sort of at the plasmid level stage where it has homology arms and a guide RNA that drive it for insertion into locus 1. And then it's got guide RNAs of other kinds to do other things. So for example, in mosquitoes, this locus in the mosquito called FREP1 is necessary for propagating, for parasites to propagate through the mosquito. So it's a host factor required for the parasite to be happy in using the host. And it's been shown that if it's mutated, then the mosquito just doesn't take the parasite very well. So you could have a guide RNA that's carried on this plasmid, this vector once it's inserted that does that. Then another one, and I'll talk a lot more about this one in a moment, is this idea of what we're calling allelic drive, where let's say you've got an allele of a gene, like the sodium ion channel, that is one that you would like to replace with a wild-type allele. So insecticide resistance comes in two flavors. One is that it can, mutations that are in ion channels, most of these insecticides, many of them, target in, the nervous system and insecticide and in ion channels. Insect-specific in ion channels. That's one of the reasons they can be relatively specific to insects and not mammals. But in any case, as of course you know, if you apply insecticides for too long, you get insecticide-resistant mosquitoes. And there's a million ways that happens. One is to mutate the ion channel itself that the pesticide binds to. The other is what's called metabolic resistance, where you have P450s and things that metabolize the insecticide that are either duplicated, have their activity changed, or a variety of combinations of those things, so that for one reason or another, the insecticide is more efficiently metabolized and destroyed. Both of these can be approached in a similar kind of way, but it's most easy to look at it from a point of view, for example, of the sodium ion channel, for which probably the most consistent and reoccurring mutation is a mutation of 1014L in the sodium ion channel to F, the 1014L to F mutation. You see it in a huge range of different mosquitoes all over the different parts of the world, all across different insects, from cockroaches to all kinds of other beetles, all other types of insects. So this is a very frequent target for mutation, and it's because the pesticides like DDT and pyrethroids, they bind to the sodium ion channel at this location, and if you make this mutation, it doesn't destroy the function of the sodium ion channel, but it renders them incapable of binding the pesticide. Okay, so that's why that works. Now, if you could make a guide RNA that cut the mutant form but not the wild type form, and you generate your stock in a wild type background, then presumably as this would sweep through the population in combination with a gene drive, then or put on a gene drive, then it should cut the other allele, the insecticide-resistant allele. It should then be repaired by the sensitive allele that the original stock carried, and then you should resensitize the insect to pesticides. It costs about $100 million or so, it's estimated, to generate a new pesticide, and another $100 million to bring it to the field. So people are estimating now that making a new pesticide and implementing it costs about $200 million. So this is a pretty inexpensive way to get a little bit more out of the existing ones that there are. Another thing that they can be doing is they can be used as another way of neutralizing a gene drive. And so if you imagine you have a gene drive that's based on Cas9 and a guide RNA that's copying itself at one location in the genome, you can have a chaser that's another. Chasers are in general anywhere in the genome that copy along with the gene drive, so they're at a second site. And this one has a guide RNA that cuts to drive it at its own location, and then two guide RNAs that cut the Cas9, so that stops the action that way. Now, the advantage of an e-chaser is it'll work on all gene drives, whether they're as long as they're driven by Cas9, while erasers have to be built and inserted exactly where the gene drive is, because the way it works is it's exactly at the same place. It cuts out the gene drive and then it copies itself into the whole. So it has to be at the same place in the genome. So erasers have to be specific for a given gene drive, whereas these do not. On the other hand, erasers have certain advantages to them too, because, like for example, in the case of the yellow mutation in fruit flies, we've made these erasers that have at the cut side, that have in the cassette a recoded version of the yellow gene. So when they cut out the gene drive element, they restore the function of the yellow gene, so that they're restorative in addition to deleting the gene drive. And that's much more difficult to do at a distance with a chaser. And of course, you know, the real simple answers, they both have flaws, and I'll show some of the data in a moment, but they both work pretty well. But probably the best thing to do is just to combine them if you really wanted to get rid of something. And then together, the hypothesis is that they would be effective. And these are experiments that are ongoing. And finally, just as a kind of genetic trickery, another way you can do things that has certain advantages, it's very helpful for experiments in the lab, but also could be used in a variety of field circumstances, perhaps, is you have a source of Cas9 that's a standard Mendelian source of Cas9, and a guide RNA cassette that's in a second location that works this in the following way. So both of these are Mendelian stocks on their own. But if you cross them to each other, what happens is this. This guide RNA cuts where the cassette is. And of course, because if you cross them to each other, then there's Cas9 that can be provided in trans. And then this guide RNA cuts where the Cas9 is. And so now, when you cross them, then from that point on, you will have established a full gene drive system, because now every single time the Cas9 will get copied to the other allele, every single time the cassette will get copied to the other allele, and then from that point on, it'll propagate. But you can raise them separately. So if you were worried about escape of a gene drive system as you build it up for release, then you could do it this way, where they're each housed separately. Nothing could escape until you wanted to, and then you'd cross them, and then they could go. Also, if you do it this way, there's more cargo you can carry along with. So you could have one set of cassettes over here, another set of cassettes over there. You'd have more you could deliver. But really, for the lab, it just serves as a very good system for studying gene drive systems. For example, what you can do is you can hold this constant and vary the kind of Cas9. So is it a Cas9 that's optimized for not cutting off targets? Is it a Cas9 that's unstable so that it rapidly falls apart after each cell cycle? Is it a Cas9 that's drug regulatable, so on and so forth? And now you can do a good comparison because you have a fixed set of guide RNAs that you can look at. We're trying to do. Now finally, I'd like just to talk about this idea of using copycat elements, which have the guide RNA, but no Cas9 that can then be driven in the presence of Cas9. So it's kind of like what that experiment that I just showed before. But in this case, it's you say you want to combine two of them that are either on the same chromosome or different chromosomes. This one has a guide RNA for where it's cuts where it's inserted. This one a guide RNA for where it's inserted. In the presence of Cas9, they cut like this, and then they copy themselves. And they can copy themselves like that whether they're next to each other or on different chromosomes. In fact, as a kind of interesting experiment that I've often suggested to graduate students in genetics is that, let's say you had two of these elements and they work perfectly. How would you map them if you didn't cheat and do DNA sequencing? And the question is, could you? And the answer is there's only one organism I know in which you can, and that's fruit flies. And the reason it's hard is that how do you map things? You map things by do they segregate away from each other? And you map them if they're closely linked. You map them if they're closely linked, then you say, okay, they're on the same chromosome. And how closely linked they are tells you how close they are to each other. But if they copy in a way that's completely independent of segregation and linkage, then how can you tell? Now the answer in fruit flies is that there's a deletion collection that spans the entire genome. So what you could do is you could take each one of those elements and cross them to the deletion collection. And when you crossed it to the deletion that was where the thing was inserted, it would only be inherited at 50% of the time instead of 100% of the time. And that way you could map the two. But that just gives you a sense for how fundamentally different this kind of genetics is than normal genetics. Okay, so this is kind of an idea, again, also with applications to crops. But if you wanted to say, make humanized models that, okay, sorry, humanized models that had many different characteristics, and you wanted to combine them, then you could cross two in the presence of Cas9, and you'd see that in the presence of Cas9 they copied each of them. And then you could combine those with another set that you'd had, again in the presence of Cas9. All the kids would get one copy, but then in the presence of Cas9, they'd get copied to all the other alleles. And then if you said, okay, at this point, I just want to stop and go back to a Mendelian sense, then you just cross two individuals that had that characteristic, and one of the chromosomes has Cas9, the other doesn't, so you can look at for the quarter that don't have the Cas9, and now you've got a completely stable stock. So now you could take these very complex humanized stocks that are, for example, humanized for the immune system, humanized for drug metabolism, that independently would be impossible to combine, right, by standard genetics. But now you just cross them to each other, get rid of the Cas9, and then you could go on doing this. And so it's sort of these modular ways of combining large arrays of genes. I just want to end also just by saying that it's very important to do these experiments safely, that they were done safely in a barrier facility, all the mosquito work as well. And these are people at UCSD that are working in all these different areas of what we're referring to of active genetics in mosquitoes, fruit flies, human therapy applications, mice, fish, plants, and yeast. And so there's a whole community of folks up there at UCSD that are doing that. And again, this was largely done by Valentino and a technician in the lab, but now we have many other collaborators as well. For those of you who might have interest in some of these projects, where we've been very fortunate to get very good support for a project to combat vector-borne diseases in India, and we are applying active genetics to a variety of other processes as well. So there's a lot of opportunity should any of you be interested in some of this. Thanks.